As we continue to look at the prokaryotic and eukaryotic cell structures, we're not going to focus on the internal features of cells. All cells contain a watery fluid, protein, macromolecule-filled environment called cytoplasm. DNA, the genetic code, is found as a circular molecule in the nuclear region of the cytoplasm of prokaryotic cells. Notice that in prokaryotic cells, DNA is circular and it is typically only one molecule. On the other hand, in eukaryotic cells, DNA as the genetic code is found as a linear molecule with multiple linear chromosomes inside the nucleus of the eukaryotic cell. Prokaryotes contain DNA outside of the single circular chromosomal DNA. This extra DNA is found as a small circular units called plasmids. Plasmids are independent of the chromosomal DNA, replicating on their own and containing a few genes non-essential to the cell. Eukaryotic cells also contain extranuclear DNA. This DNA is found inside an organelle called the mitochondria. The mitochondria contains its own genetic material in the form of one circular DNA molecule. In addition, in photosynthetic microbes, DNA is also found in the chloroplasts, also as a single circular molecule. Let's now look at the site of protein synthesis, the ribosomes. In eukaryotic cells, ribosomes are found in two places, free in the cytoplasm or attached to the endoplasmic reticulum. This endoplasmic reticulum is now called ROF-ER or RER. Ribosomes are structures made of two components, a large subunit and a small subunit that assemble together to make the entire ribosome. In the case of eukaryotic cells, the large subunit has a measurement unit of 60 Sedberg. The small subunit has a measurement of 40 Sedberg units. Once these two subunits are assembled together, the entire ribosome will have an 80 Sedberg units of size. Sedberg units are units made to measure ribosomes depending on their sedimentation rate, their molecular weight, shape, and other criteria. Ribosomes are also found in the mitochondria of eukaryotic cells. Ribosomes in the mitochondria of eukaryotic cells also contain two subunits, a 30S small subunit and a 50S small subunit, which once assembled make up a 70S ribosome. Ribosomes are also the site of protein synthesis in prokaryotic cells. Ribosomes are found free in the cytoplasm of prokaryotic cells. Prokaryotic ribosomes also are made of two subunits. The small subunit is a 30S subunit. The large subunit is a 50S subunit. Once assembled together, the ribosome has a 70S unit size. Another internal structure in both cells are the structures where energy is synthesized. The site of ATP synthesis in eukaryotic cells are the mitochondria of the cell. In the prokaryotic cell, the cell membrane is the site of ATP synthesis. Some bacterial cells can form endospores. Endospores are dormant forms of the cell. Endospores are tough, resistant structures that help microorganisms survive extreme environments. Helping microorganisms survive extreme heat, like lack of fluids, radiation, etc. On the other hand, eukaryotic cells, fungal cells, can also make spores. Eukaryotic spores are found in fungi. However, the spores found in fungi are reproductive structures. Fungal cells can reproduce sexually, producing sexual spores, or fungal cells can also reproduce asexually, creating asexual spores. You should now be able to answer the following questions. Which internal structures would make good targets for antimicrobial drugs? Remember that a good antimicrobial drug must target specific and relevant structures, giving the drug good selective toxicity. Antibiotics that target prokaryotic ribosomes can be more toxic to humans than other groups of antibiotics. Why would an antibiotic that binds to a 30S or 50S ribosomal subunit harm eukaryotic cells? Think of a specific organelle found in the eukaryotic cell.